Welcome back, Gen Bio 1 folks, to Unit 4. And in this chapter, or in this unit, we are going to be covering chapters 9 and 10. However, we are going to cover chapter 10 before 9. And my reasoning there is I like to make the sugar before we burn it. So I like to do chapter 10 before chapter 9. Let me say a little bit about this chapter or this unit of information. This is a chemistry heavy chapter, and it has some pretty difficult concepts in it. So you are going to want to spend a lot of time, and the best way that I can suggest is just do the same thing over and over and over and over. It's really just spending a lot of time with the material. There are tons of videos loaded in the unit, in the module, in Canvas. And so I encourage you to watch those over and over and over to help you comprehend this material. As usual, don't hesitate to contact me if you have any questions. And if I have enough emails, I'll set up some sort of study session. I may also try to run a review session before the next test. So stay tuned for that, or send me an email if you would be interested in that. So chapter 10 covers photosynthesis. And photosynthesis is a process by which solar energy is transformed into chemical energy. Photosynthesis converts carbon dioxide and water to carbohydrate. Now think about that for a minute. Carbon dioxide and water, and you make sugar. Okay, plants have the ability to do that, and we're going to look at the details on how they are able to do that. So not only plants undergo this process, but some photosynthetic organisms called algae, which are in kingdom Protista, also undergo this process, and some bacteria can also undergo this process. So here's some pretty pictures of photosynthesis. And here's another picture showing the diversity of organisms that can undergo photosynthesis. And this is found on page 188 of your books. So if you recall from the last unit, the organelle of photosynthesis is the chloroplast. And the chloroplast is found in plants and some algae. Now keep in mind the bacteria do not have chloroplasts, but they can still undergo this process. Some bacteria, not all bacteria, some bacteria. So the chloroplast is the organelle of photosynthesis inside the eukaryotes, inside the plants and the algae. The chloroplast can be divided into two parts. The first part is called the stroma. And the stroma is the semi-fluid interior of the chloroplast. It is in between something called thylakoid membranes and the two outer membranes of the chloroplast. And at the stroma, what happens is this is where sucrose, starch, and other carbohydrates are assembled. And I'm going to refer to this set of reactions as the light-independent reaction of photosynthesis. The light-independent reaction, meaning that it does not require light in and of itself to undertake that reaction. The second part of the chloroplast is called the thylakoid membrane system. It is the inner membrane system of the chloroplast, and that is where light energy is converted to ATP. And this is referred to as the light-dependent reaction. So this set of chemical reactions is dependent on light. And the thylakoid membranes contain molecules that absorb sunlight or the pigments. And so here's a picture of some cells that are capable of undergoing photosynthesis. This is a cross section of a leaf. So if you were to take a leaf up to your eye level and hold it flat and to look at it uh, just right in front of your eyes, this is a cross section of the leaf. If you take a look at the picture, okay, you'll notice that there are some horizontal arranged cells on the top. That is the upper epidermis of a leaf. The bottom edge of the leaf isn't quite so clear, but there's a layer of cells similar to that on the bottom edge of the leaf. And then in the middle of those two layers of epidermis are the cells that are responsible for most of the photosynthesis. And they are green and they're packed with chloroplasts. So you have a picture similar to this, or this exact picture on page 189 of your books. So again, if you take a look, the cross section of the leaf is on the left, and you'll see in white the upper epidermis and the lower epidermis of the leaf. And then in the middle of that are the photosynthetic cells. And you blow that up and you see all those green dots inside that cell, and those are all chloroplasts. And then those chloroplasts appear green. If you were to take and dissect a chloroplast, in the upper right, you'll see that's what the chloroplast looks like, kind of cut open. And in the upper right is the cartoon version, and in the lower right is kind of an actual photograph. So the stacks of green coins are called the thylakoid membranes, and this is where the light-dependent reactions occur. And then the jelly-like fluid surrounding those green membranes is called the stroma, and this is where the light-independent reactions occur. And again, that's located on page 189 of your books. So in order to have a good understanding of what's happening and how light actually gets converted to chemical energy, we have to have an understanding of the visible light spectrum. And this is pictured on page 192 of your books. And if you take a look, okay, you have the whole electromagnetic spectrum here in gray, that bar across the top. The white part is just the visible light spectrum, and this is what our eyes can see. If you take a look at the gray part, the electromagnetic spectrum, our eyes cannot see radio waves, they can't see microwaves, they can't see X-rays or UV light. So our eyes are only designed to see the visible light spectrum. The visible light spectrum you'll see goes from about 380 to 750 nanometers. And you'll notice that it's divided up into colors. Does anybody see a pattern in that coloration? If you go from right to left, you will notice that it's the colors of the rainbow. And a good way of remembering that is Roy G. Biv. And Roy G. Biv stands for red, orange, yellow, green, blue, indigo, violet. And those are the colors of the rainbow, and hence the visible light spectrum. And here's just another picture of that, kind of the same information. But again, going from right to left, from a longer wavelength to a shorter wavelength, we go from red, orange, yellow, blue, green, indigo, violet. So let's talk a little bit about those pigments and how they apply to photosynthesis. Different pigments in plants and other organisms that undergo photosynthesis absorb light wavelengths at different spectra. So for example, we have chlorophyll, which is the pigment that's most commonly known. And chlorophyll absorbs the spectrum in the violet, blue, and red, but it reflects green light. It is the main pigment in green plants, and that is why plants appear green to us. And we'll talk more about that in just a minute. Another group of pigments are carotenoids, and those absorb wavelengths in the violet to blue spectrum, but they reflect red, orange, and yellow. And there are certain vegetables that we eat that are high in carotenoid pigments, such as carrots, yellow peppers, red peppers, squash. All of those are high in carotenoid pigments. And then a third group of pigments, which is not as well known on land and are not talked about in your book, but are a group of pigments called phycobilins. And phycobilins reflect the wavelengths of red and blue. And phycobilins are found in some red algae and some bacteria. So plants, the leaves appear green to us because they are reflecting the pigments of the green plant. They are absorbing all the other colors of the spectrum, but they reflect green. And that is what happens most of the year. But in fall, what happens is the days get shorter and shorter. The plants have ability to pick up on this. The chlorophyll is not able to undergo photosynthesis as long as it usually would, and it starts to break down. And when the chlorophyll breaks down, then you start seeing the other pigments in the plant come through, such as the carotenoids. And that is why in the fall we see leaves turning red, orange, and yellow. It's because the chlorophyll has broken down and the leaves are turning red, orange, and yellow to our eyes. And so that's what we're just demonstrating there. Is why are leaves green? It's because they're absorbing all the other colors of the spectrum, but they're reflecting green. And so here's just a good look at if you were to graph out the wavelengths at which the different pigments absorb light. You'll notice that there are two main chlorophylls, chlorophyll A and B, but there's actually 5 or 6 different chlorophylls now. Chlorophyll A and B are certainly the most well studied. And then you'll see the carotenoids as well. And this is talked about and pictured on page 194 of your books. And so here's some fall color for you. And again, the chlorophyll has broken down in many of these trees, and so they are reflecting the carotenoids, the red, orange, and yellow pigments. And so let's talk about the overall reaction. This picture is not in your book for some reason, but there is a similar equation on page 191 of your books. And I encourage you to spend some time with this reaction. You are going to have to know it. So we start out with 6 molecules of carbon dioxide and 6 molecules of water. And in the presence of light energy, those 2 molecules in light energy undergo a process called photosynthesis. And the end result of that is 1 molecule of glucose, which is C6H12O6, plus 6 molecules of oxygen. This is a chemically balanced equation, which means if you count all the C's, H's, and O's, you will have the same number of carbons, hydrogens, and oxygens on either side of the arrow. And let's review a little bit. What kind of molecule is glucose? It has carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. Therefore, it's a carbohydrate. Secondly, you can tell it's a carbohydrate because glucose ends in OSE. So I encourage you to watch this BioFlix photosynthesis video. It's very good, and I encourage you to watch it many times, and it will be also on your homework in mastering. And I believe it's also loaded in Canvas in the module as well. So here's just another look at that. This is pictured on page 191 of your books. And so this gives us a little bit more detail showing that that equation that we just saw is going to be a multi-step equation. And so if you take a look on the right, we've got water and sunlight. And we are going to end up with sugar and oxygen coming out of this process. And this process is going to be a little bit more detailed than we saw just in that equation. So in order to understand how this happens, we do need to talk about reduction oxidation reactions, also known as redox reactions. And this is discussed on page 165 and 166 in your books. So in oxidation, a substance loses electrons. So if a substance loses electrons, it is oxidized. In reduction, a substance gains electrons or is reduced. In other words, the charge is reduced, so it's gaining an electron. And so this is what happens in the form when we mix sodium and chloride and it forms table salt. What happens is each of those tends to become an ion, as we talked about in the chemistry chapter, is that sodium tends to lose an electron and it gives that electron to chlorine. So the fact that it lost an electron, sodium is the substance that is oxidized, and chlorine takes on that extra electron, so it has been reduced. And this is in chapter 9, page 165, 166 of your books. But this is talked about on page 191 of your books in chapter 10, and it talks about the substance that becomes reduced in photosynthesis and the substance that becomes oxidized in photosynthesis. So what happens is carbon dioxide gets reduced and water becomes oxidized in the reactions of photosynthesis. So here's a nice summary that's a good way to go back over and over and over. And let me see if I can find the page number for you on that. I don't have the page number, I apologize on that. That may not be available in your book. But there's a very similar picture back on page 191. In fact, that's essentially the same picture as page 191. It just looks, the drawing is a little bit different there. So let's talk about the reactions of photosynthesis. And this is a summary. We're going to go into more detail. But we're going to start with the light-dependent reactions. And remember, those occur at the thylakoid membranes. And two subsets of the reactions include a set of reactions called photosystem 2 and photosystem 1. And the products of those reactions are then going to derive the light-independent reactions, which we said happen in the stroma of the chloroplast. And that set of reactions is commonly referred to as the Calvin cycle. So what you may want to do, what may be helpful, is to take this slide and print this out. This is kind of a good big picture slide that you can kind of have over you as you're poring over the details of photosynthesis, and you can go, okay, I'm going back, and this is the light-dependent reaction, and this is photosystem 2 or photosystem 1, and you can kind of keep things straight in your head. So let's go and talk about these reactions. We're going to start with the light-dependent reactions. And remember, that's going to be photosystem 2 and photosystem 1. The end result of this reaction, or these sets of reactions, are ATP and NADPH are formed. And those are two of the molecules that are going to drive the next set of reactions. So let's talk a little bit about them. We've talked about ATP. Remember, it's a nucleic acid. But let's talk about NADPH because we haven't talked about that, and I'm not going to have you know what NADPH stands for, but just know that NADP is a coenzyme. A coenzyme is an enzyme helper. It carries electrons, and so when it receives an electron, it's NADPH. When it doesn't have that extra electron, it's NADP. So the light-dependent reactions are the first stage of photosynthesis, or the photo part of photosynthesis. And just as a review, these occur in the thylakoid membranes of the chloroplasts. So let's get an idea of what happens actually when light hits those chlorophyll molecules. If the pigment is not part of a bigger system, the electron can't stay in the outer orbit. But what happens is that electron, when the light hits the chlorophyll molecule, that electron gets pushed up, it gets boosted to a higher energy level. And that is a very unstable situation. And if it can't stay in that outer orbit, it falls back to its original orbit. And when it falls back to the original orbit, the electron releases energy as heat and fluorescence, or fluorescence in the form of this light change. Okay, so let's go over the reactions, the light-dependent reaction, and remember that photosystem 2 happens before photosystem 1. It's just kind of related to the fact that we're doing chapter 10 before we're doing chapter 9. So photosystem 2 happens before photosystem 1. And this picture is pictured on page 196 of your books. As you go through this, if you start getting confused and you need to go back to the big picture, there's a good summary picture of both photosystem 2 and photosystem 1 on page 197 of your books. So let's take a look at this pigment, and these are, there are separate pigments going on here, and these pigments are part of a photosystem. And those photosystems can be divided up into two parts. If you take a look at this picture, the overall big part is the purple part, and that's called the light harvesting complex. And then inside that is the lavender part, and that's called the reaction center complex. So light hits the pigment, we have the chlorophyll inside these photosystems, and the light is absorbed. So the energy from that light is added to an electron. That excited electron leaves its normal orbit and jumps to an orbit further from the nucleus of an atom, just like we were talking in the previous slide. So instead of that energy being released as fluorescence, the energy is passed to another pigment and then to another pigment as it goes through this light harvesting complex. Eventually, it gets passed to a special pair of chlorophyll A molecules. And eventually, from that pair of chlorophyll A molecules, that electron, the energy is passed to a primary electron acceptor, and that's that lavender square in the reaction center complex. So finally, the energy is transferred to one pigment of a pair of pigments in the reaction center complex. Okay, so we're going back a little bit. We're going to these P680 molecules, the two green balls. And then, so I'm repeating what happened in the last slide. And then that P680 pigment gives up its excited electron to the primary acceptor, which is the lavender square in the reaction center complex. Okay, so then an enzyme splits a water molecule into two electrons 2 hydrogen ions, and an oxygen atom. And this is what we refer to as splitting water in photosynthesis. The other electron will do the same after another P680 molecule gives up an excited electron. So remember, we have 2 green balls, those P680 molecules. That happens twice for 2 electrons. So have all the electrons of photosystem 2 been accounted for or replaced so the system is ready to run again? Yes. So now that electron from photosystem 2 passes through an electron transport chain, and that is made of a molecule called a plastoquinone, PQ for short, and it is just an electron carrier. And it involves also a protein called plastocyanin. So while in the electron transport chain, the electron's energy is used to pump hydrogen ions into the thylakoid lumen, that's inside the thylakoid membrane, making a concentration gradient. And you can take a minute and watch this video. It's helpful to keep things figured out. So the hydrogen ions are allowed to diffuse down their concentration gradient through a protein. This powers the protein to synthesize ATP from ADP and a lone phosphate group. So remember, we're going back to ADP plus P gives you ATP. And remember, ADP is the uncharged form and ATP is the charged form. And so here's kind of a summary of what we've just been talking about. And this is pictured on page 198 of your books. Now we haven't finished, we haven't gone through photosystem 1 yet, but I just kind of wanted to give you the big picture of what is going on with photosystem 2, with the PQ and the PC, and where we are in the big picture of things. So here's just an analogy between the photosystem 2 and photosystem 1. On the left is that photon from the light exciting that electron and what happens, and they eventually, through a series of complex reactions, that electron is then being used to make ATP. And this is pictured on page 198 of your books. And this is just a good description on how the thylakoid membrane converts the light energy to chemical energy. And this is actually described in more detail in chapter 9. This is the only part where it's a little bit of a problem with covering chapter 10 first. But this process is happening both in respiration in the mitochondria, and it's happening pretty much the same way in the chloroplast that we're talking about now. So you got to cover it one way or the other. So, but if you are struggling with this whole process of how the light energy going to chemical energy to ATP and NADPH, you can go back to chapter 9 and it might describe it in more detail for you. And this is pictured on page 200 of your books. So let's take a look at the photosystem review. Photosystem 2. We still haven't done photosystem 1, but let's talk about what's happening in photosystem 2. What are the beginning components of photosystem 2? Sunlight and water. What are the products of photosystem 2? Oxygen and ATP. Is photosystem 2 endergonic or exergonic? It's endergonic because you have sunlight and heat driving that reaction. It requires energy. And what does the light do to a pigment electron? It excites it to a higher energy shell. So where is that energy transferred? It goes to excite another pigment electron. And where does that final electron go? It goes from one of the pair of P680 pigments in the reaction center complex to the primary electron acceptor. Remember, that was in the middle of the molecule. Where does the electron come from to replace the electron lost by one of the P680 pigments? From an enzyme splitting a water molecule. Review, what type of molecule is an enzyme? It's a protein. Another quick review, what are the building blocks of proteins? Amino acids. Let's continue with this photosystem 2 review. The electron from the primary acceptor travels down the electron transport chain and powers a protein to do what job? That protein pumps hydrogen ions into the thylakoid membrane. These hydrogen ions then diffuse back out of the thylakoid and power a protein to make what molecule? ATP. What is the job of ATP? To transport energy from one part of the cell to another so another molecule can perform work. Remember, ATP is not good at holding energy, it's good at transferring energy. Have all the electrons from photosystem 2 been paired back with another atom? No, one loose electron has excited the electron transport chain. And there's that picture again showing the whole process. And again, that's pictured on page 196 of your books. So let's go way back to the beginning, it seems like. The overall reaction of photosynthesis. Okay, we are halfway through the light reactions on the left. And you can see there, we've got two things going into that. We've got light and water. We have oxygen being released. We're going to have ATP and NADPH being made, but we're not there yet. We still have to go through the second part of the light reactions. Okay, so let's go back. We're in photosystem 2, and in photosystem 2 we made ATP. Now we've got to get to photosystem 1. Remember, we're still in the light-dependent reaction. So the energy from the sun, what we originally started with, also powers a protein to make a NADPH. Remember, in photosystem 2 we made ATP. Now in photosystem 1 we're going to make NADPH. And like ATP, NADPH is another molecule that transfers energy to molecules that use energy to carry out cellular processes. So thinking about that, is this reaction exergonic or endergonic? It is endergonic because it requires the input of energy. So in the same process as photosystem 2, in photosystem 1, light excites an electron in one pigment, and this energy is passed from pigment to pigment in the light harvesting complex until it is passed to one of a pair of pigments. And in this case, remember, photosystem 2 is P680. Photosystem 1, that pair of pigments is going to be P700. And that is in the reaction center complex inside the lavender box there. Just as in photosystem 2, the excited electron is given from one of the P700 to the primary acceptor. So at this point, are there any holes where electrons are missing? Yes, one electron is missing from 1p700. This hole is filled by the electron from photosystem 2 that traveled down the electron transport chain. So we have the excited electron from photosystem 1 is passed down its own electron transport chain through a different protein called ferredoxin, or FD for short. This electron and one more from another round of photosynthesis 1 are transferred to NADP molecule using an enzyme to make NADPH. Okay, so on the right-hand side there you can see it's very similar to photosystem 2, just different molecules involved. So we go back to the big picture with photosystem 2 and photosystem 1. And again, this picture is on page 197 of your books. And you'll see photosystem 2 on the left with the P680 molecules, photosystem 1 on the right with the P700 molecules. What comes out of photosystem 2 on the left is ATP. What comes out of photosystem 1 on the right is NADPH. And here's just even a simpler version of that. It's the same picture, and this is pictured on page 198 of your books. So let's go through a recap of photosystem 1. What is the beginning component? Sunlight, just like photosystem 2. What is the end product of photosystem 1? NADPH. What are ATP and NADPH? Compounds that transfer energy to molecules. What does the light do to a pigment electron? Excites it to a higher energy shell. Where is that energy transferred? To excite another pigment electron in the photosystems. Where does the final electron go? From one of the pair of P700 pigments in the reaction center complex to the primary electron acceptor. And remember, that's in the middle of that photosystem. Where does the electron come from to replace the electron lost by the P700 pigment? From photosystem 2. Remember, we have an extra electron that comes over from photosystem 2 that drives photosystem 1. The electron from the primary acceptor travels down the electron transport chain and ends up attached to what molecule? NADP. How many electrons must be attached to NADP plus to make NADPH? 2. Are the chemical reactions of photosystems 1 and 2 endergonic or exergonic? They are endergonic because they require the input of energy, which is sunlight. Where do photosystems 2 and 1 take place? In what organelle and what structure inside the organelle? They take place inside a plant cell, and inside that plant cell, inside the chloroplast, and inside the chloroplast, inside the thylakoid membrane. Okay, so if you're not confused enough, we're going to confuse things even a little bit more. Focus mainly on that photosystem 2 and photosystem 1, and remember that occurs in the thylakoid membranes of the chloroplast. But keep in mind that's a generalized process, and some organisms, like the bacteria, have a variation of this process. They will only use part of photosystem 1, and then they don't use photosystem 2 at all. Other organisms use both photosystems and the cyclic electron flow. But in this process, light excites a pigment electron whose energy is passed from pigment to pigment and then to a P700 molecule and then to the primary acceptor. Then the electron is passed to ferredoxin, which delivers it to a cytochrome complex where it is used to make ATP. The electron is then transferred to plastocyanin, which transfers it back to the hole left in P700. Don't get too caught up in this whole process, but this is just to show you that there is a variation in the system. Normally, can photosystem 1 take place without photosystem 2? No. And so here's a picture of that cyclic electron flow, which is a variation of photosystem 1. But like I said, I really want you to focus on getting that whole, the whole photosystem 2, photosystem 1 process down, and then just look at this as just a slight variation in the process. And this is pictured on page 198 of your books. So this wraps up half of photosynthesis. Okay, this is the photo part of photosynthesis. We still have the synthesis part of photosynthesis to do in the next PowerPoint. Okay, so we have wrapped up the light-dependent reactions. This is what happens when light hits a chlorophyll molecule. Okay, next up will be the light-independent reactions, or the synthesis part of photosynthesis.